Thank you, Jonah, for this opportunity, and thanks to Texas Parks and Wildlife for the funding and the opportunity to present the research as well. And thank you for all of you coming to hear about this research about the Nelson's pocket mouse in Texas as well as beyond. So let's get started with introducing you to our rodent here. This is Chaetodipus nelsoni. This rodent is found in the family Heteromyidae. We might be familiar with some more charismatic members of this family, like kangaroo rats and their relatives. This genus, Chaetodipus, as well as another one, Paragnathus, are smaller, more quadrupedal representatives within the family. Common name for this group is often referred to as spiny pocket mice, and our particular species, the Nelson's pocket mouse, tends to have a wide elevation and distributional range throughout its distribution, although it does have a habitat preference of rocky habitats. We'll be getting back to that later. Similar to other heteromyid rodents, the Nelson's pocket mouse is nocturnal, it's granivorous, which means it eats seeds, and according to the International Union of Conservation, it is of least concern. So no concerns currently in terms of the longevity or extinction risks for this particular species. What I've shown here is the distribution of this rodent in Texas. And hopefully you can all see, I don't know if you can see my mouse or not, but there's a shaded region primarily in western Texas where Chaetodipus nelsoni is known to occur. This map comes from the Schmidley and Bradley Mammals of Texas book. And every place where you see a black dot is a known county record where we actually have specimens in collections for this particular rodent.
And Jessica, just to cut in, we can see your mouse, so it works to use it as a little pointer.
Great, I'll use that. Thank you so much, Jonah. Now, something that's a little bit interesting about this distribution is this record down here of Webb County, Texas. It seems to be a little bit spread apart, far away from the other records that we're seeing in Texas, and has led some to wonder if this isn't a possible outlier. Is this really a true population of where you can find these rodents? And part of that comes from just our geography of Texas and where you find particular regions and habitats. So for example, both of these maps are indicating in the blue line the Balcones Fault Zone. And what we often see is a pretty discrete difference in the fauna that you find west of this fault zone as well as east of the fault zone. Some species, sure, we can find all across the state, but many times this fault zone can differentiate or be the, say, western limit for some species and the eastern limit for other species. The figure on the left, I've circled again this distribution of our pocket mouse, where the majority of our samples are in the Trans-Pecos region of Texas, but then we have this one Webb County specimen down here in the Rio Grande Plains. These are different ecosystems, different types of vegetational areas, different types of habitats. And you can see that really well with the picture of the map that I took on my office wall the other day, where through this Balcones Fault Zone, or at least part of it, you can definitely see there's a big difference in what you see in terms of habitat, precipitation, climate that is east of that line, as well as what you see west of that line. And recall that our mouse of interest, the Nelson's pocket mouse, prefers rocky habitat. We're definitely going to see that more west of the Balcones Fault Zone versus east. So this type of habitat differences that we see on either side of the state definitely calls into question whether or not there is a population of mice way down here in Webb County, Texas, especially knowing that they have these habitat preferences for rocky habitat. Let's talk a little bit more about what we know about the specimen records from Webb County. First of all, there is only one museum specimen in collections, only one. It was collected in 1969. We'll see that on a later slide. And this specimen was originally housed at Texas A&I University, which was Texas A&M Kingsville. A few years ago, I worked with Dr. John Baskin, and we did a permanent donation of the specimen to the Biodiversity Research and Teaching collections here at Texas A&M. So we now have this rodent in our collections up here in College Station. The specific locality Where the specimen came from is generally referred to as Hilltop Oasis. This is west of Encinal, and it is at the intersection of Texas 44 and US 83. The general area where this animal came from was about 0.66 kilometers in perimeter and is generally rocky. Here we have a Google Earth image of what the area looks like. So here's our intersection of 44 and 83, and our rockier habitat can be seen all within this area. And it is surrounded, I just want to point out, surrounded generally by green, lush habitats. Lots of it is also privately owned and agriculture as well. If we got on the ground and we're walking around in this general area, these are some pictures of what the habitat looks like. So it is kind of rocky and sparsely vegetated. So maybe there is this population of Chaetodopus nelsoni in this little tiny region of Webb County, if not more dispersed than that. What's interesting further about this record, not only of it just being one specimen, but also possibly being potentially constrained within this one little region around these two highway intersections, is if you try to look for the nearest specimens in collections. To the north, the nearest specimen's about 150 miles away. That's up in Valverde County. To the west, the nearest specimen in Mexico is about 70 miles away. So this looks like a truly isolated population potentially, that or no one has ever really looked for additional specimens. So given its isolation, given its time of collection, it leads us to a few questions of, is there really a population of Nelson's pocket mouse down here in Webb County? Or maybe, is there even additional populations along this stretch shaded in gray? Are there more Chaetodopus nelsoni here and we just haven't collected them into museum collections? Another alternative, given again the date of collection, the location of where this specimen is located or was located relative to other specimens, perhaps this is an extinct population that no longer exists. Lastly, there's always the potential of species mix-up. First, I will say that this specimen was morphologically examined. It is definitely a Chaetodopus nelsoni. So it is not another species of Chaetodopus, like say the hispid pocket mouse Chaetodopus hispidus. But we do know, if we look at the collection notes, the field journals of the collectors that picked up this particular specimen, we know they were up in Valverde County collecting 2 days later, based on specimens that they collected and installed into collections. So is it possible there was some sort of mix-up? Maybe, might be difficult to tell. The status of Chaetodopus nelsoni in Webb County is certainly concern for conservation, because if there is a population here, it may be isolated from other populations to the north and to the west in Mexico. And this could be a concern of identifying a lineage that might be in need of conservation. So trying to solve this problem of what is going on with this particular species down here in Webb County is of interest for conservation. So we wanted to ask the broad general question, thanks for funding from Texas Parks and Wildlife, is Chaetodopus nelsoni in the Rio Grande Plains of Texas? Do we find it down in the southern portion of Texas and east of the Balcones Fault Zone? We took two approaches to try to address this question. The first one was with mapping and the second one was with fieldwork. So first I'll get into the mapping. First, I should say that none of this work goes on with just one person. This is certainly a team effort. We have pictures here of graduate students as well as undergraduate students who participated in various parts of this research. I will be listing out their names at the very end of this slide, but you will see their pictures throughout the presentation for the different sections of where they contributed. So with the mapping, again, this was with funding from the State Wildlife Grant through Texas Parks and Wildlife. We took two approaches here. The first one was using Google Earth. And basically, instead of spending hours and hours and days on the field, in the field, walking through properties, we thought, since we know we have a rocky habitat specialist, that we could use Google Earth images using this Webb County location sort of as our guidance or our model, and look for regions that look like that, all the way from Webb County, Texas, up to Valverde. and see if we can find large enough sections of land where the animal could live and identify them for possible trapping later. So Layla especially spent a good bit of time looking at maps, again, using this Webb County location as a model, and she found 126 potential sites spread across 4 counties where maybe the habitat would be appropriate enough for the Nelson's pocket mouse. Once she got on the ground with a team of workers, that 126 potential sites dropped very quickly down to 3 potential sites spread over 3 counties, just due to the size of the area, types of vegetation, what that rock habitat actually looked like. I will get back to the results of those 3 potential sites later, but I do think it was a really efficient use of time to examine the land area via above grounds before actually getting on the ground to set traps for these animals. The second bit of mapping that we did was ecological niche modeling. I'm not going to go into a lot of detail here, but what we were trying to do was to determine the probability of species occurrence in a particular area. And you use a variety of data to help you do that. So for example, you will use records from natural history collections. Where has this animal been found before? You'll also use habitat variables like habitat substrate, temperature, precipitation, all downloadable fields that you can get through BioClim and other resources. And what this image on the bottom is showing you is an idea of what that kind of looks like. So we have species occurrences, and these occurrences are records in natural history museums. And then we have several layers of information, whether that's temperature, maybe some sort of element concentration or salt. You put them all together in environmental space, and then you build a habitat suitability map. Adrian and Layla worked on this part, and this is what our habitat suitability map for Chaetodipus nelsoni looks like. So seen here is a map that includes Texas as well as the United States, Mexico, as well as the United States, primarily Texas and New Mexico. And where you have dark colors indicates low suitability in terms of habitat. So low likelihood of species occurrence because the habitat suitability is not that great, especially using records from museum collections. The lighter we get, the more yellow we get, indicates high habitat suitability, high likelihood of species occurrence. If you look at our outline of Texas here, you can see that, okay, we have some regions of high occurrence or likely high occurrence in the Trans-Pecos region, but as you go down this western edge along the Rio Grande of Texas, we don't really have a lot of yellow coloration going here. So this seems to indicate that the habitat in this area may not be appropriate for Chaetodipus nelsoni to occur. So again, our work thus far, just doing some mapping in museum records, is indicating that maybe this region of Texas is not the best in terms of habitat suitability for our species. That takes us to our next step of actually getting on the ground and doing some field work to see if we can find the Chaetodipus nelsoni throughout this area of Texas. We actually did this a couple of different ways. With funding from Texas Ecolabs, we also wanted to explore a little bit in the Valverde and Edwards counties. We had 6 properties total in these regions. These are private properties that we had permission to trap on. And Layla, Grace, and Emma spent some time out at these properties. And I'll cut to the chase right away. We never caught Chaetodipus nelsoni at any of these properties, despite the rocky habitat and possibility of species occurrence there. Where we spent most of our time were in Webb counties, as well as the 3 counties that Layla had previously identified as probable good habitat locations, and those were Dimmitt, Kinney, and Zavala counties. We did this work all primarily through the funding of Texas Parks and Wildlife, and get to some of our methods of what this field work looked like. We primarily did this work in the spring and summers of 2018 and 19. Again, 4 counties covered. 3 of them were primarily indicated through Google Earth, as well as ground searching of indicating that the area looked good enough for trapping. But we spent most of our time at the locality where Chaetodipus nelsoni was trapped initially back in 1969. In total, 22 nights of trapping, over 6,500 traps were set, and over 400 animals were captured. However, none of them were Chaetodipus nelsoni. For fun, what did we catch? Lots of other things belonging to two different rodent families, the heteromyids, again, those are the kangaroo rats and their relatives, as well as the chrysetid rodents, which are pretty common throughout the state. I don't have common names here, but I'll run through them briefly. In the Heteromyidae, we got the hispid pocket mouse. We got a kangaroo rat. We did not identify that down to species. We released all these animals. So I'm skimming over the methods. In the previous slide, I'll go back actually one. These are live Sherman traps. They're baited with sunflower seeds and oatmeal, and they allow us to release non-target species. Good thing, because that's all we caught. So we released all of these, which meant that sometimes we couldn't identify animals to species specifically. We also caught Perignathus, which is a pocket mouse, and in the Chrysetidae, common names going down, Baomys taylori is the northern pygmy mouse, Neotoma is a wood rat, grasshopper mouse, and Nicomedes. White-footed mouse, most likely Peromyscus leucopus. We also got fulvous harvest mouse, as well as hispid cotton rat. Now, what's interesting about all of these captures is that generally do not have strong preferences for rocky habitats. Additionally, research has shown that Chaetodipus nelsoni, our target species, generally avoids sandy soils and disturbed habitat. So these captures, as well as our lack of capture for Chaetodipus nelsoni, can indicate that the habitat where we caught all these animals, although it may look rocky to us, is not rocky enough for Chaetodipus nelsoni, at least potentially. So what these findings, both the mapping as well as the fieldwork, seem to indicate, especially knowing that this mouse collected in 1969 is a legitimate Chaetodipus nelsoni, but it seems to indicate that perhaps there is no longer a population of Chaetodipus nelsoni in Webb County, Texas, or along this western edge of Texas in the Rio Grande Plains. It's pretty hard to prove absence, though, so it's hard to come down definitively to say, like, nope, it's not there. It may never be there anymore. We shouldn't expect to see it. So I would recommend continued trapping, or at least looking for the specimen when opportunities provide themselves. So looking for the species when possible. Because again, we have these conservation implications that maybe there is a very small population of Nelson's pocket mouse down in this part of Texas. And if there is, that would definitely be consideration for conservation. We're going to come back to this mouse in Webb County in a little bit. But what I wanted to talk about and branch out to now is the fact that Chaetodipus nelsoni doesn't live in a bubble of Texas, right? It is found in other areas, so beyond Texas as well, including New Mexico and into Mexico. The map that is shown here is from the Mammalian Species Account published by Troy Best in 1994. What it is showing is that we have two subspecies, a northern subspecies and a southern subspecies. Origination of the species likely occurred in northern Mexico, so the majority of the distribution of the species is along the Mexican Altiplano. And there are some differences between the northern and southern subspecies. There's some coloration differences, there's some differences in terms of the skull and shape of the skull, and there's also some karyotypic or chromosomal differences as well, which often can indicate that there might be cryptic species in the area. Furthermore, within this region, there are a number of well-known potential geographic barriers for species distributions. This is a figure from a manuscript that was published last year, specifically on this species, where we laid out some of these potential barriers. So one is the Rio Grande River. Two is along the mountains, so Sierra Madre Occidental. We also have a really well-known filter barrier that divides a lot of taxa. Number 3 down here is the Southern Coahuila filter barrier. And lastly, another barrier up here, the Rio Conchos. what's interesting is if we went back to the previous slide and looked at those two subspecies, they're actually distributed across these different potential barriers. And knowing that we have some morphological differentiation as well as karyotypic differentiation could point to the possibility of cryptic species or hidden, hard to observe species within Chaetodipus nelsoni. And we wanted to pursue that question in greater detail. Are there cryptic species within this lineage, especially given that little bit of differentiation that we're already seeing morphologically and karyotypically, as well as the presence of these potential distribution barriers? So what we did was a full-out phylogeographic assessment. We obtained specimens, morphological as well as genetic specimens from all the way across the range of this species, from up in New Mexico down south into San Luis Potosí and other states within Mexico. We gathered 4 different types of data, including mitochondrial genetic data, amplified fragment length polymorphism data, karyotypic or chromosomal data, and morphological data. I'm not going to go into any of the detail of the methods, but I would be happy to talk about those after if anyone's interested. In total, we looked at 470 specimens from 142 localities, including some peripheral new populations like the one in New Mexico and some a little bit further west than had been previously recorded. What we found, to summarize briefly, is general support for 3 distinct lineages: mitochondrial DNA supported, AFLP supported. There are karyotypic differences across this range, as well as some morphological differences. Morphology didn't end up being something that we could rely on in terms of always having distinct morphological traits that we would see in particular areas. It was a little bit messy. And we think a lot of that had to do with the ecological variation in general. We're seeing this lineage of rodent across very high or very differentiated elevational gradients, as well across a pretty large geographic range. So that can come with plastic or variable morphology. What we recommended in our work is the elevation of the 2 subspecies up to subspecies and a recognition of a 3rd species. So in total, what we end up having is Chaetodipus nelsoni down here where the type locality of the species is. To the west, we have a new species, Chaetodipus durangi. And to the north, we had support for 2 subclades, which we're recognizing as subspecies, Chaetodipus collis mapimiensis and Chaetodipus collis collis that we find in Texas. Make sure that worked. Okay. Some of the things that this tells us in terms of conclusions or general findings about Chaetodipus nelsoni across this range is that there is continued support with this species for cryptic diversity in the Mexican Altiplano. This region tends to be rich in terms of species diversity, and this is yet another example of finding that with our Chaetodipus nelsoni group. What's interesting in terms of possible geographic distributions is that it seems for this particular species that drainage basins, river drainage basins, seem to have more an impact of species presence and differentiation than some of the other filter barriers. So for example, the Southern Coahuila filter barrier, which is here, number 3, going across, kind of cutting west to east, didn't really have much of an impact for our species at all, even though it has very large impact for other species. It does seem that the rivers, at least particular areas of the rivers, are serving as potential limits to geography for some of these species. And at this point, it's a little unclear if the Sierra Madre is also serving as a potential filter, because what is indicated over here to the left is we have some specimens that we were unable to get sufficient data from. We have morphological data from these specimens, but we don't have molecular data. So we're not exactly sure to which group they may be located in, and if these filter barriers or other potential geographic barriers can be affecting their distribution. So, of course, this research has led to unanswered questions. One of them being those specimens that we only had morphological data for, and I'll get back to that in a second. But a first question that we had stemming from this research has to do with the status of Chaetodipus lineatus. This particular species is distributed wholly within the distribution of Chaetodipus nelsoni. And it has been described in the literature as being pretty similar to the Nelson's pocket mouse, but maybe just not having those rump spines. So something I neglected to say earlier is that our species morphologically is one that is often defined as a spiny pocket mouse, or at least having spines on its rump in different parts of its body. There are smooth-haired Chaetodipus as well, and they tend to break up in terms of relationships. So this other species, Chaetodipus lineatus, seems to look just like Chaetodipus nelsoni, but it's lacking some of those spiny hairs. There have been multiple attempts by multiple researchers to trap and find Chaetodipus lineatus based on its original descriptions, and all of them have failed. Past research has often shown that it's also very unlikely that you would find sympatric, coarse-haired species. So it's unlikely to find overlapping distributions of these Chaetodipus that are more coarse or spiny in their hairs. So it seems a little questionable of, does Chaetodipus lineatus even exist? Especially because we know that juvenile specimens or molting specimens often lose those spiny hairs. So is it possible that the specimens in collections are actually just Nelsoni missing those spiny hairs? So that is one unanswered question. The other one has to do with those specimens for which we lack molecular data. We don't know which species or subspecies they belong to. This has implications for biogeography. And this also includes our Webb County specimen that we can get back to from the beginning of the talk. So current ongoing research, this is not published, is taking an attempt to address these two unanswered questions. And it seems like, gosh, we'd be awfully limited because we really need molecular data to do this, right? The best way to get good, clean molecular data is to have fresh tissues. Well, when you have specimens that were collected in 1969 or maybe earlier, most folks weren't collecting tissues at that time. All we're left with are skin, skull, and skeleton material in natural history collections. However, with the advent of various new techniques, molecular laboratory techniques, it's very possible to get good molecular data from museum skins. So what we did is requested a bunch of specimen loans from museums and specifically asked for a digit to be cut from a forelimb. These don't really have much utility in terms of morphological examination or phylogenetic examination, and we can get a good bit more DNA out of a bone than, say, a snip of skin tissue or hair. So this is referred to as destructive sampling of the museum specimens. We were successful getting that from, I think, about 3 different collections. And we worked in a designated ancient or historical lab here at Texas A&M where we only have older samples of specimens coming in. That's going to help prevent contamination with more recent samples that maybe we're getting from fresh tissues or things like that. Then we tediously work in small fragments, and this is still a rather antiquated type of methodology, but this is what we did. We worked with very small fragments in the lab so that we would have overlapping regions of the gene that we're interested in. Nowadays, more advanced methods involve things like next generation sequencing, where you can use short fragments pretty easily. That's how the work is designed, and short fragments are pretty much all that we have in our historical museum specimens. So anyway, that was a long way to say we could get molecular data from these specimens, and we could try to answer these 2 unanswered questions about lineatus and then specimens lacking molecular data. So first we'll do lineatus. In total, I had 12 museum specimens from which I was able to get good DNA. This included the type specimens of Chaetodipus lineatus. The type specimen is where that species was originally described. These 12 specimens came from 7 localities across San Luis Potosi. And genetically, what we found is that every single individual grouped firmly within clade 3 of the previous Chaetodipus nelsoni research, and that is now recognized as Chaetodipus nelsoni. So this molecular data strongly supports that Chaetodipus lineatus is not a valid species. And it's likely those specimens that have been designated as lineatus or described as lineatus in the past were probably young or molting Chaetodipus nelsoni. And we might need some additional morphological work to get some eyes on those specimens to get a better idea of that. But in general, this molecular work is strongly supporting Chaetodipus lineatus is not a valid species. How about those specimens within the nelsoni complex that were lacking molecular data? In total, we had 18 museum specimens that I was able to get DNA from 9 localities, all in Coahuila. And nearly all of them grouped with clade 2B, which is Chaetodipus colis mapimiensis, all except this southern locality right here, which actually grouped down with Chaetodipus nelsoni. So this work is going to allow us to increase the range of nelsoni up a little bit further north. And it's going to allow us to increase the range of this colis subspecies further to the east. So how about our Webb County specimen? It's right there. Wouldn't it be great if everything worked well in the lab? I had difficulties with this specimen. I had multiple extraction bouts that I did, different digits each time. Very careful in the lab to make sure that I didn't have contamination, which I did not. But I was just never able to get really clean sequence data. The DNA was just never that great, slightly degraded. But if I looked at things like how genetically differentiated was the sequence that I got from this specimen from, say, colis up here, colis in Texas, as well as colis mapimiensis to the west, what I found always was that there was a closer genetic similarity of our Webb County specimen to Chaetodipus colis mapimiensis to the west of it. This gives us some further findings in terms of geographic or potential geographic barriers. So for example, now I think we can say that this second potential barrier, which were the Sierra Madre Occidental, the mountains over here, are not serving as a barrier to distribution for Chaetodipus nelsoni species. The other thing that's kind of interesting is initially we thought the Rio Grande was serving as a barrier, which it is between 2 subspecies, but with this Webb County specimen grouping more closely with specimens to the west than specimens to the north, this tells us that perhaps the Rio Grande didn't serve as much of a barrier for this particular population in Webb County. Maybe there's some different structures to the river down there and what it looks like, how wide it is, what it looked like in the past, to facilitate movement of Chaetodipus colis mapimiensis across the Rio Grande in this area. So some of our overall findings from this work, which includes the work that I presented earlier, the first part of this talk about Texas Parks and Wildlife funding, as well as work that we were doing on the side with some collaborators, is that now instead of 1 species, Chaetodipus nelsoni, we're recognizing 3 species within this complex, colis, Durangi, and nelsoni. We've also found that Chaetodipus lineatus is likely not a valid species, bringing the total number of Chaetodipus species down to 19. And updating the numbers of both smooth and coarse-haired forms that we find. In terms of our Webb County specimen, assuming again that there were no mix-ups in terms of collecting and that there was a population of animals in Webb County at some point in time, data seem to indicate that it's likely that a population no longer exists there, nor anywhere along the Rio Grande plains of Texas. Again, absence is a difficult thing to prove, so additional work couldn't hurt here, but we do have our additional findings with the mapping work that was done, the ecological niche modeling, and we have some biogeographic work in the broader context indicating that Chaetodipus colis is not coming down north to the south. There is a big gap here, at least an apparent gap in distribution. Lastly, instead of having Chaetodipus nelsoni in the state with our updated taxonomy, we have Chaetodipus colis, again, likely totally restricted to the Trans-Pecos region and likely not west of the Balcones fault zone. As I wrap up, I would just like to acknowledge my collaborators on the larger Chaetodipus work, so taking Chaetodipus nelsoni into a larger framework across its full distribution. This includes Brett Riddle, who you can see here, Sean Neiswender, who is not working, but he's sitting with his traps, and then David Hafner, a longtime collaborator of mine since I was in graduate school. And of course, we'd like to thank all the students of mine that have participated in this research. Their names are indicated down to the right, everyone who participated in mapping as well as fieldwork, and Texas Parks and Wildlife for funding. Thank you all for your time, and I'd be happy to take any questions.
Thank you, Jessica. That was awesome. I have several questions already. And again, as a reminder, if people are uncertain about how to ask questions, look for that little box in the lower right-hand corner for the chat box, and you can type in your questions there. So first question was by Chris Kloss, and he said, have you considered, or have you thought of the potential that this was an individual or small group that was displaced by a Rio Grande flood event?
Ooh, it certainly is possible. How we might be able to rule that out, I might open my share screen again, is that if it was a Rio Grande flood event, I guess we're assuming then that the flood event would have brought populations from the west over the river to the east, unlikely to have been a flood event down from the north to the south, right? So at least based on our findings about where our specimen genetically places. What I'd like to do to be able to support that more is see if there's any good historical information about flood events, but also see if there's any other additional like relictual populations or specimens that are in that eastern part of Mexico closer to Webb County. Similarly, is there any possibility of finding any additional specimens through that Rio Grande Plains of Texas? So I think it's definitely a valid hypothesis, and we just need some more data to help address that. It's a really interesting question.
It's a neat idea. I mean, some of those big giant floods could have huge amounts of debris floating down, and I can imagine like it could have even been a population that lasted for a decade or two or something as a result of a historic flood event. It's just an interesting idea. Mm-hmm. Cliff Shackelford wrote, and he said, just a comment, Nelson, who this rodent was named after, was a powerhouse biologist. He has many accolades, including exploring the Sierra Madre, where he encountered and collected the now extinct imperial woodpecker. And Cliff is our state ornithologist, by the way, for Parks and Wildlife. And then Michael Wardner said, what would the conservation status of C. Colis colis be now, given the reduced range, G and S ranks? I don't know. That's a question you can answer without running it through the NatureServe rank calculator, but it definitely would be different than it is, I think, considering the whole population altogether.
Yeah, and I think you have to take it in a broader context and include New Mexico as well, because there is that population up there that was previously unknown. And I forget if it's a historical population or if they're actually animals up there isolated. So taking that subspecies as a whole would need that information, and probably also how are we going to, well, yeah, actually that would be what we would need to take into account, is that New Mexico population with that definition for that subspecies.
Mm-hmm. Lee Fitzgerald said, thanks for the presentation. It was a great project and I enjoyed hearing about it. So that was just a kudos.
Thanks, Lee.
Michael Warner wrote again and said, any threats that we're aware of?
I don't think so. These are, seed-eating rodents, rocky habitats. If anything, maybe with climate change, as things dry up, maybe they'll be okay because of their preference for rocky habitats, but then that's going to also follow their vegetation preferences as well and the types of plants that they prefer. So if climate change affects the distribution of those plants or potentially causes their extinction, the rocky habitat may be there, but the food resources may be gone.
It does seem like they're primarily found in parts of the state with relatively low human populations?
Oh, that will always have an account various factors as well. In the paper published last year by Nice-Winter et al., they did have some comments and cited some literature about how the species doesn't necessarily do well with disturbance. So it does prefer more of these native habitats. You're not going to find it likely being able to survive with humans for very long, if at all.
And then Evelyn Mers wrote and asked, what effect on population distribution do you think climate change might have? Name of the feline assistant. Oh, she also wants to name, was there a photo of a cat in one of your, or do you have a cat there with you?
I don't know Layla's cat's name.
Okay.
Yes, so yes, I can ask and find out for you if you would like. Actually, Layla just recently put that up. If she's still on, because I know she was on earlier, she could potentially type in the Q&A the name of her cat. But yeah, I stole that picture recently because I thought it was great, and her cats do assist with everything. What was the question though about distribution changes? Nova. Okay.
I just wrote. Yeah. But, and then she said she was asking about climate change, which I think you mentioned. And we were welcome to elaborate on it more, but I don't know that we have data.
It's always so hard to predict. We haven't done any of that research yet. We certainly could, model habitat changes into the future. Folks are doing that 20, 50, 100 years into the future, but we haven't done that for this particular species complex yet.
And then Michael Warner said, what might be the next rodent species issue to address? That's for you, Evans. Winky face. Michael used to be my boss over at Parks and Wildlife before he went to the dark side and works at Fish and Wildlife now. So we've recently worked on Texas kangaroo rat, and that species continues to get a lot of attention due to its consideration for federal listing action. The other one that we worked on not too long ago, or I believe that work is still continuing. We're trying to figure out what the next steps are. Was with Sigmodon fulvinter, right? That Caleb is working on at Texas Tech. Is that right?
Yeah, they've been presenting that at the Texas Mammal Society meetings, Texas Society of Mammalogists. And I'm not quite remembering the details of it right now, but I think the general question is does it exist? Is there still a population of it?
It's quite similar to this one. There was a historic location. They went and looked for it, and similar to yours, they have not found it yet, and they're kind of wanting to continue work looking for it to see if they can increase their sampling effort and get closer to that idea of, is it absence or did we just miss it? We got another question from George Oliver. Is human agency a possibility, say transportation of the Webb County animal or a gravid female with livestock or other goods from Coahuila?
Sure.
Stumbled on that. Yeah.
Yeah, it's just one of those things that can certainly exist as a valid hypothesis, but extremely difficult to test, right? Unless, I guess a good way to test it now is to get a mouse and throw it in a car and see what happens, kind of thing. But we don't want to do that. So yeah, I think it's certainly possible that there could have been some transports into Texas from Western Mexico somehow. Yeah.
Then Adrian Castellanos, I don't know if you pronounce that with the
Castellanos, yeah, Adrian.
Okay,
yeah.
Any ideas about the importance of river basins versus other barriers, shifts in vegetation or soil, competitive exclusion by other species?
Yeah, that touches on multiple things. So it certainly seems like the work that was published last year on the species complex as a whole, that the river basins have a very large effect for this particular species complex. And the basins probably have effects for other species as well, where at the same time, some and not all of those possible geographic barriers are having much of an impact. one of my take homes from that is the importance of treating species individually, right? And not just assuming, oh, these basins have an effect on this species, so they probably have an effect on everything else that's co-occurring in the area.
Mm-hmm.
And that may not be the same just because a particular species requirements for survival. And also their ability to live within an ecosystem and potentially competing with other, in the case of Chaetodipus, granivorous or seed-eating mammals. So Chaetodipus in general, and I need to go back and review a little bit of this genus, but you don't find many species in sympatry or co-occurring with each other. So it does seem like that there is probably some competitive exclusion going on, or some limiting similarity in some effect of where one species will prefer one particular type of niche and another species another one. So I'd previously mentioned there's these smooth-haired and coarse-haired forms of Chaetodipus. Those can occur in sympatry because they are differentiating their niches and how they're using them. But you don't usually find smooth hairs together, and you don't usually find coarse haired together. So I think I got all of Adrian's questions with that one, but I'm not sure.
You know, I think that seems to be similar with kangaroo rats, where there tends to be smaller kangaroo rats and larger kangaroo rats. And you tend to find sometimes one of each in an area, but not generally are there two large kangaroo rats overlapping or multiple small ones overlapping.
Yep.
I mean, sure, there's plenty of exceptions to that, but just in my experience, when I'm looking for kangaroo rats, it seems to be that there's either one small one and one large one, or one of each, or whatever. But, okay, so I think we got one more question here. Michael Warner again asked, what other projects is Dr. Light working on? If you're working on some other ones right now.
I could stay focused on Texas because that may be the preference for this particular crowd. With a couple of collaborators that I think have joined the call as well, we're working on some Peromyscus research. Maybe they're not all the same. Maybe there's some differences going on across the full distribution. Maybe we actually might have some separation of habitat for some of our Peromyscus species here in the state. So very broadly, we're looking into some Peromyscus stuff. I really like working with gophers, pocket gophers. So quite a bit of pocket gopher ectoparasite research going on, led by a graduate student in my lab. And then through collaborations, I'm doing a variety of other mammal work as well as parasite work. A lot of it always centering on museum collections and the importance of collections and research. Certainly the work that I presented today, we couldn't have done that without museum collections. So one of my major pushes, which I'll be following up on through the years, is pushing the importance of museum collections and how we can be using them in our collections in this, or in our research in the state and beyond the state as well.
Not to mention the bear paper that we worked on together.
Yes.
Which is
yeah.
Getting closer slowly to.
Yeah. Jonah's talking about a paper about the distribution of black bear in Texas and using a combination of natural history museums and citizen science, both records supplied to Texas Parks and Wildlife, as well as citizen science observations on iNaturalist to try to update the distribution of the species in the state. And we're hoping that that will be coming out in Southwestern Naturalist in 2021.